I humbly in, I humbly invite all the dignitaries, those who have joined from our state as well as outstate of Gujarat. So I welcome all of them on behalf of our SIPS family. So I am here to introduce our today's personality, today's eminent speaker, Dr. P.D. Bharadia sir. So actually, sir doesn't need any introduction. Sir is very known personality in field of pharmacy, but the student needs the introduction. So that's why I am here to introduce him. So Dr. Praful D. Bharadia sir is currently working as a professor at Department of Pharmaceutics and Pharmaceutical Technology at the reputed College of India, LM College of Pharmacy. Sir did his BPharm and MPharm from LMCP only. He completed his MPharm in 1988. Sir is having vast experience, 30 years of vast experience. Out of this 30 years, 23 years of teaching experience, 5 years of research experience, and 2 years of industrial experience. So during teaching, Sir has worked with some organizations like K-Biper Gandhinagar, S.K. Patel College, Ganpati University, B.S. Patel Pharmacy College, then Indore Institute of Pharmacy, Ratnamani Pharmacy College, and currently working at LMCP. So under guidance of Sir, 11 PhD scholars awarded their PhD degree, and Sir has already filed 9 patents, and out of this 9, 2 is already granted. Sir published 5 books as author and co-author. Sir is having around 110 national and international publications in reputed journals, national and international journals. Sir has done around 35 presentations in some national and international webinar, workshop, and seminars. Sir has also got some awards. Like first award he got is Indian Achievers Award for Education Excellence in 2010, presented at 26th Indian Achievers Summit on the challenges of economic growth and social development, 22nd November 2010, Delhi. Then second award he got is Rashtriya Gaurav Award in 2010 for outstanding contributions, services and achievement, presented at Delhi. Third award he got is Indira Gandhi Shiksha Shiromani Award in 2013 for outstanding achievements in field of education, presented at New Delhi. Sir has associated with some associations, and he is having some memberships, like he is currently working as an honorary secretary of IPA-Gujarat state. He is visiting faculty of NIPER Ahmedabad. He was dean at faculty of pharmacy Ganpati University at 2005. He is core committee member of centralized placement cell GTU. He is currently working as a SSIP coordinator LMCP, and he is currently working as a member of board of study of GTU. So we have very appropriate speaker with us. Sir is having very good knowledge. I also studied under his guidance. Sir, I am very much delightful to have you with us. And I remember that you were my guide in BPharm, and I learned a lot from you. And I have done the project on engineering side somewhat, but automation in our pharmaceutical industry. But I learned a lot during BPharm also. So thank you very much for accepting our invitation, and we heartily welcome to you on behalf of our SIPS family. Thank you very much, sir. Please.
So can we start?
Yes, sir. Thank you so much, Ankit sir. Yes, sir.
Okay. It's a very nice afternoon today, and I'm happy to see so many people, so many young minds gathered today. So it's going to be a good experience for me also to share some of the issues which I thought would be interesting. I thank Dr. Ankit for inviting me, and he told me that, sir, could you spare some time for talking to the students? I said, for students, any time is available, and you know, you can ask me anytime. I'll be the pleasure to talk to the students because that's where I always love to be with, So it's really very fascinating to talk with students, to work with them, and to learn from them also, right? Because these young minds are wonderful. They have so much of ideas. So it's nice to connect with you all. So I thank Ankit Bhai, the management, Saraswati Institute, and the faculty members over there, the students, and interestingly, our old colleague Dr. Padmanabhan and his colleagues are also here from his college. Other faculty members and students from other institutes also who would be in this webinar. Uh, it will be a, because the topic which I've chosen is challenges, So we are trying to, we'll throw up so many challenges during this session, and we will try to, uh, make some effort to understand these challenges because there is a big gap. There's a big gap in technology, and the gap is largest in parents. If you see other dosage forms, tablets, we are pretty conversant with tablet medicine in the college and all that, the IPQC test. But in case of parentals, the gap, technological gap is very high. Therefore, many students, when they go to the industries, they face a shock that we have not seen anything like this, we have not learned anything like this in college. So it takes time for them to adjust to that environment. So I'll just tell that even when I passed out my master's and I was thinking that uh, I would like to go into manufacturing, learn the intricacies of manufacturing, and then shift possibly to the field of choice. So I was thinking which dosage form department I should join. It was a question which was there in my mind also. So looking to that, then I said, okay, which is the most difficult area to work in? So obviously we came down to parentals. Parentals is an area which is most difficult. So I said, okay, let me try to work in that area of parentals and try to learn something. So it was an interesting journey. And during my course of presentation, I may share some experiences also which I have learned and I have encountered during working in the industry also, and our exposure in that time, this whole journey. So with your kind permission, I can share the presentation.
Sure, sir. Sure.
Okay. My voice is audible?
Yes, sir. Presentation also visible.
Okay. Fine. I'll make it full screen. Is it fine, everybody?
Yes, sir.
Okay. So the topic which we are trying to dwell on today is challenges in parental processing and bridging the gap. I will be trying to stick more into the processing part or the technology part, and I will not dwell more into the formulation because formulation itself is a big topic. So if you go to combine everything, it may not be possible in the scope of this discussion. So wherever possible, I'll just, if required, I may go to some formulation aspect, but mostly I'll be talking on the processing part because that is where the gap is more. Right. So going to the introduction part of parentals, as we all know, the prominent methods to deliver drugs are oral and injections. These, as we know, they are the most popular methods of delivery. In orals, we may be having tablets, capsules, and liquid orals. Most of the drugs have been formulated to accommodate the oral or the injection delivery routes. If we go to see today, new biological drugs such as proteins, peptides, nucleic acids that are currently parenteral require novel delivery technologies that will minimize the side effects and lead to better patient compliance. So this is something which is going to come in. It is already there because we are seeing these days that if you see the last few years, the blockbuster drugs, if you just Google it, that top 10 selling drugs in the world, at least 7 to 8 of those 10 would be proteins and peptides. These are all patented molecules and they are blockbuster drugs. Therefore, and if you go to see, most of them are parenteral in nature. Okay. Therefore, we are looking at a delivery system which is always associated with new drug deliveries and these days biological in nature. Therefore, the interest in parenteral formulations is renewed, maintained, and is being increased. And then we are coming forward with newer formulations also. It's not just a simple injection, but in that case, in parentals also, we are having so many modified drug delivery systems which are now being developed. So it's very interesting and fascinating. So why parentals? We all know benefits of parental systems are major. In case of any emergency cases, in case of trauma, if you want more bioavailability, that is always because of parental systems. If you see most of the patients who are hospitalized and under medication, then mostly they are being given parentals. If you go to any ICU in a hospital, rarely you will see any other dosage form being administered. Most of the medicines are parenteral in nature. So for critical conditions, always this is the preferred dosage form, as we all know. There are certain limitations of parentals, as we all know, that it has to be sterile, non-toxic, non-pyrogenic, and if there is any contamination, it may lead to death of a patient also, right? So we cannot withdraw the dose if it has been administered. You need a skilled caregiver to administer the dosage form. So these are things which also go with these parental systems. Obviously, suitable drug characteristics should be there so that they can be formulated into a injection. The drug, we know that we always check the pre-formulation characteristics, whereby we see the solubility and everything about the drug. So that properties are always essential to study to get a safe, effective, and stable dosage form. Stability is also important because if the composition of the formulation is not correct, then the formulation will not be stable, right? So that comes into formulation. The routes of administration becomes important because this is from where you are starting actually. It's very interesting that when you are going to design an injection, you would first see what will be the route of administration. The route of administration decides the rest of all the planning of formulation also. Because you have, how do you decide that whether you're going to prepare a 5 ml injection, a 2 ml injection, or a 10 ml injection, or maybe a 0.5 ml injection? That is a very nice question which one has to decide. That how do you come to a decision that when you are having a new API with you, how will you prepare the injection, in what volume you will deliver it? You know the container which you're going to use. Say you may be using a vial, or you may be using an ampule, or you may be using a prefilled syringe. But the question would be, what would be the volume? Whether I should prepare it in a 0.5 ml volume, or I should prepare it in a 2 ml volume or a 5 ml volume. So the decision of that would be taken primarily on two things. One is the solubility of the drug. If the drug is easily having aqueous solubility, then any amount of drug you would be able to solubilize in a small amount of vehicle. But if you are going to administer the drug through a subcutaneous route or an intradermal route, then you have a limitation of volume, right? So your very important formulation would start with route of administration, because the whole formulation route or the process would depend on this and the molecule characteristics or the API characteristics. So that is the importance of route of administration, because we know that there are various routes of administration available. We'll see one more slide. If you go to see, briefly I'll go through all this, that what are the types of parenteral sterile products available. So if you go to see, we have solutions. If you go to see the pharmaceutical classification, then we have solutions, suspensions, emulsions, dry powder injections, freeze-dried products. These are some categories of formulations which can be given in parenteral form. Solutions are the most popular, easy to administer and easy to make. You may have if the drug is insoluble in water, you may have a oily injection, or you may have prepare a suspension. But when you're preparing a suspension, one has to be careful that you will not be able to give it intravenously. It has to be an intramuscular suspension, right? Then you have emulsions also, but in case of emulsions, unless it's a nano emulsion or it is an emulsion that's a TPN, that's a total parenteral nutrition. Only these types of emulsions or nano emulsions can be administered intravenously. Otherwise, the rest of the emulsions have to be administered also intramuscular. Dry powder injections, if you go to see, then they are prepared to be reconstituted before it is administered. So you always are provided a water-free injection container along with the dry powder injection. The drug is in powder form. It is dissolved inside the injection bottle or vial by administering water-free injection, and then it is administered. This is required specifically when the stability of the drug is less, or the drug or the API, which we call, is easily hydrolyzable, which means in presence of water, the drug is degrading. Therefore, you will not be able to prepare a liquid injection because drug will degrade. Therefore, you are preparing the injection in dry powder form, and then that dry powder form injection is being administered. Before administration, it is reconstituted in water-free injection. So that is why you will see dry powder injections in the market. Basic reason is because of stability issues. The freeze-dried products are also there. In case of many such products which are high value and which are such that they cannot withstand typical processing conditions of sterilization and all that. In such cases also, you may have freeze-dried injections available. Many vaccines and high value injections are also like immunological products. So many, if you go to see, then these are all prepared in freeze-dried conditions. So you may have dry powder injections, you may have freeze-dried products. Both are in powder form. Both have to be reconstituted before administration. But the freeze drying is carried out for high value products. Again, where there is more stability issue, you may go for a freeze drying of the product, right? So that is where there's a slight difference between dry powder injections and freeze dried products. So these are different categories of the pharmaceuticals which are in injection form. Similarly, you may have biologicals. You may have biologicals, and these biologicals are there like vaccines, toxoids, tissue extracts, biological extracts. These are also sterile in nature, parenteral in nature, because they are given by injection form. So these biologicals, we are very familiar with the vaccination drive which is going on, the COVID vaccines which have been developed popularly, two products, Covishield and Covaxin, and everybody across the world is taking these vaccine shots. They're all again parenteral in nature and given intramuscular. Okay. Are also parenteral in nature and they are being administered. Allergenic extracts are also in injectable form, so they are also parenterals. Radiopharmaceutical products are also there for many times you want to do cancer detection and all that. So radio contrast agents are injected and then the scan is done. So in such cases also, these are parenteral products. Then other products like dental products. We have local anesthetics for dental tissues. Many people, as you know, as you grow old, there's a problem in the teeth and there may be dental caries. You may have to go for root canal. For all these procedures for removal of tooth, the procedures always, there's a local anesthetic which has to be administered. These are all parenteral injections which have to be used. Biotechnology products like monoclonal antibodies, they're also parenteral in nature. Ophthalmic products, again, a big category where you have eye drops, eye ointments. These are also parenteral in nature. Though these eye drops and eye ointments are for external use because we consider eye as an external product organ, yet these products are parenteral in nature or sterile in nature. Why? Because they are supposed to be sterile, since if they are contaminated, eye being a very sensitive organ gets an infection very fast, and therefore all the products which are supposed to be administered to the eye are also sterile. Okay, medical devices. These things are not in front of us, but they are used every day in and out in operation theaters in the hospitals. Surgical reserves, syringes, catheters, endoscopes. These are all, things which are used, catgut, sutures, ligatures, surgical dressings. These are all sterile. So it's a big, big list of products which is supposed to be sterile. Each and every product has its own properties, and therefore we know that we select a different method of sterilization and processing depending on the requirement. I'll not go into the methods of sterilization all, but wherever required in some of the examples when we take, we'll try to touch upon two major methods which are being used. Okay. Routes of injection, parenteral administration. The most popular I've written over here like intramuscular, subcutaneous, intravenous, and intradermal. These are the four most popular ones. Then we have intra-arterial, intrathecal, intracisternal, intrapleural. These are all other specific routes of administration depending on the requirement of the patient and the disease for which the injection is to be delivered, right? So according to what we have said, that depending on the route of administration, the volume which is to be administered is decided. Accordingly, the injection is formulated. So we'll see a few examples of non-sterility. So these are real examples, and from that we learn from these case studies, and therefore we come to an idea what can go wrong and what we have to take care of. Right, these are some case studies. There was a Devonport incident in UK. Post-operative patients were administered infusion fluid, dextrose, 5% dextrose, very commonly used. 7 people were administered and 5 out of them died. So they stopped the administration because there was something very serious which was happening. 5 out of 7 patients died who were administered this dextrose injection. The manufacturing was by Evans Medical, which is a major UK producer. Big company like here we have Cadila, Torrent, a major company in UK. They did some investigation and it was found to be contaminated with Klebsiella aerogenes and the gram-negative coliform bacteria. It was infected by a gram-negative coliform bacteria and Klebsiella aerogenes. And approximately 1/3 of all the bottles from the batch were non-sterile. This was something very interesting, that when the batch was checked, 1/3 of the bottles were non-sterile. Now, another interesting fact was that contaminated bottles were not detected by end product sterility testing. This is the problem, because we all know that when we are testing the quality of the parenterals or injections, sterility testing is one of the major tests. Then we have pathogen testing, which is secondary, like that. So we have a list of tests which is always done on all the products. But what is being seen over here is that the product was prepared in the company. It had passed everything, all the tests which were done in the company. So it was tested for sterility also. It was passing the test for sterility. The product was in the market. After it was administered, there were problems which were seen, which means the contaminated bottles were not detected by end product sterility testing. So this is a major challenge, and we all know why this happens. It's very common because we do not test all our units, injections. We call it a unit, right? Each unit which is produced is not tested for sterility. Or we all of us have gone through the pharmaceutical monographs for sterility testing, and we all know that in table number 1, which is one of the most fascinating tables, it gives you that what is the number of units you have to test for sterility. Therefore, if your batch is very big in size, 2% or 20, whichever is less, is specified in the monograph. Therefore, you would end up testing only 20 vials or 20 ampoules for sterility from the whole batch. So the risk is very high, or the probability of getting a non-sterile unit in that 20 vials which are taken for testing is very high. So you may not be able to detect a non-sterile vial from the whole batch because we are not testing all of them. That's a big challenge. Therefore, sometimes it happens that when a shift is going on for production, it's an 8-hour shift. Then maybe the whatever product was prepared in the first 4 hours is sterile. Then after the break was there was a problem in the environment, or there was a problem in the machine, or something went wrong, and the sterility was compromised. So the rest of the product which came out was non-sterile. The QC person had come, he had taken the samples in the morning and he had gone for analysis. So in the afternoon part, all the vials which came out which were non-sterile were not tested for sterility or any other test. Therefore, many small such errors happen, and because of that, we are not able to detect a non-sterile vial in the market or the batch which is going out. We are whatever we've tested, that shows that it is sterile, so the product is passing, and the batch is released, it goes into the market, right? So this is a big challenge. Even today, we are not able to manage this issue because we rely on end product sterility. We don't have any online process where we can do non-destructive testing. So so many students are over here, and I would throw this challenge to you all, that can we develop a non-invasive or a non-destructive method of testing sterility and a non-destructive method of testing photogenicity in any ampule or in any vial? It would be of tremendous use to the industry. Can we develop such a technology? If you have any ideas, you can think about it, make some teams, apply for some funding from SSIP, we will fund it. It is going to be a real big boon to the pharma industry because you will be saving a lot of lives, like this what is shown over here. Okay, going ahead. So investigation was done, fault was detected in the autoclave. You understand? See over here. So there were two temperature probes in an autoclave. We are using steam for sterilization. So these uh, bottles of dextrose were being sterilized by autoclaving. So the exposure conditions were 116 degrees for 30 minutes, and two temperature probes were there. One at the steam inlet, there was a dial thermometer, and one at the chamber drain, which was a recording thermometer. So there were two probes. The problem which was seen was the drain chamber recording was being ignored, and the cycle was operated on the basis of only dial thermometer. So the dial thermometer was showing the right temperature, and the cycle was operated on that. After the cycle was over, the injections were removed, thinking everything is sterile. But the other sensor which was there in the drain chamber, that was ignored, and because of that, what was happening was that since proper venting was not done, so air was not removed before this pressure cycle, steam inlet was from the top, so the top bottles in the top part of the autoclave were sterilized, the bottom in the lower part of the chamber were non-sterile. If they would have checked the drain chamber recording of the thermometer, then that would have shown less temperature than 116. And as a result, the batch should have failed at that point, but they had ignored that, and as a result, this problem had taken place. Another incident, Rocky Mount incident in USA. See, I'm trying to cover one example of each type of issue. So here in this first case, we have seen that if the SOPs are not followed properly and the autoclave is not operated properly, then the products can fail. Another thing is patients were administered again here 5% dextrose water fire injection normal saline affected at least 378 patients in 25 hospitals across US. 40 patients also died. Again, contamination was enterobacter. Investigation was done. The cap liners which were used in the bottles were faulty. So seepage of cooling tap water could readily permeate inside the bottle. So when the cycle was over, they used to spray tap water inside the autoclave for cooling the bottle so that they can be taken out fast and they can be taken for packing. Now, what is the problem? When anything is hot, it has high pressure inside. As soon as you cool it, the pressure inside will go down inside the container. And when the pressure goes down inside container, there's a slot, maybe a sort of vacuum which is being slightly created. If the seal is not correct, then it will pull something from outside. So since the water was available outside, which was non-sterile, it was sucking in that non-sterile water into the container, and as a result, the bottles were getting contaminated. So, what is the key learning over here? Components are also key factors in achieving certainty. So, therefore, here we come to some idea that whenever we are going to use components or we are selecting components for a particular parental product, the vendor audit, as it is said in GMP, we call it vendor audit. Vendor audit means the person who is supplying the components to you, you have to go and inspect his premises. When you inspect the premises of a person who is supplying you rubber stoppers, when you inspect the premises of a person who is going to supply you empty vials or empty ampoules, you go and inspect a vendor who is manufacturing aluminum seals and is supplying it to you for a product which you are making. Because you don't manufacture all this, you just formulate the product and you fill it into containers and you use containers and closures which you are getting from outside. So the quality of the containers and closures is also important. So if you do vendor audit, you come to know at what level of quality your suppliers are working. And if you find that they are not working with good quality attributes, then there is a risk to your product also, because the components are not good. So that's a key learning over here. Then another one is, it's an eye ointment. Post-operative infections after ophthalmic surgery. 8 patients developed Pseudomonas aeruginosa infections. Now this is very common in India, I tell you. We said that there's a camp for cataract operations, very commonly done in villages and small centers. We have camps for cataract operations en masse. So many people are going there and they're having their eye surgery being done, right? And it has been seen many times that people lose their eyesight after the operation. Why? Because of infection. This is a similar example that after the ophthalmic surgery, the medicine which was poured in the eyes of the patient was not sterile, and as a result, they developed infections. This case was such a case was also observed in one of the hospitals in Ahmedabad, which is by the name of Nagari Eye Hospital. It's an municipal corporation run hospital near Mithakali in Ahmedabad. Similar case was observed over there before a few years, where many patients lost their eyesight because of this such a infection which was done after the surgery. The product which was administered was not sterile, and as a result, the whole problem happened. Therefore, what happened over here? It was a hydrocortisone eye ointment containing neomycin and amphotericin. So investigation was carried out. The ointment was not terminally sterilized. Aseptic manufacturing was used. No preservatives were added. Now this is an ointment, mind well, right? So the petroleum base was dry heat sterilized in bulk prior to adding of the soluble ingredients, but a film of moisture had condensed on the surface of this petroleum, and this was contaminated by Pseudomonas aeruginosa. As you know, when you want to sterilize an oil, if oil is a vehicle or petroleum or some such thing base, then you cannot use autoclaving. Why? Because when you do autoclaving, steam is going to enter into that product and it will cause problems. So we cannot autoclave any oil. Oils have to be sterilized. Oils or ointment bases which are hydrophobic or they are oleophilic, they have to be sterilized by dry heat in a water oven. Okay. So it was also seen that the Pseudomonas aeruginosa, this is interesting. The Pseudomonas aeruginosa was found on the towels, shoes, gloves, and hands of the people who were engaged in the manufacturing of the ointment. See, this is how the investigation is done. It's very interesting that they also checked that since the product was failing, they checked that in the manufacturing, is there any problem? So they checked the environment, they checked the AHUs, they checked the air sampling, they checked all the records of cleaning. They also did swab testing for the people. And when they checked, they said on the shoes, gloves, and hands of the persons who are manufacturing the product, they found this contaminant. What does this mean? Personal hygiene and training of manpower is essential. If your people are not trained properly, then they can contaminate the best product also. If you do not train them to follow the right SOPs, then also your product can fail. Though it's a good product, it can fail because of poor personal hygiene and poor training of the people who are employed or who are engaged in this manufacture. Another reason could be antibiotic resistance must have developed over a period of time in the premises. This is interesting. Antibiotic resistance, which means when there is a contaminant in the area, in a clean area or in an aseptic area, you have an SOP in place. You are doing the cleaning every day with a disinfectant. Say, for example, you're using Lysol, or you're using Dettol, you're using Savlon, or anything like that, T-Pol. With the required concentrations also, you're using this disinfectants, and yet you find that the contamination is still there. The reason is, for a long period of time, the organisms, they become resistant. They do not die, though you are using the same concentration of disinfectant. Therefore, from these issues in CGMP, it has been recommended that you should not use one single disinfectant every day. There has to be a cycling of disinfectants. That 1 week you use one disinfectant, second week you use another disinfectant. So there is to be a cycling of disinfectants, and because of this, the resistance will not develop. So this is all from the learning of these examples. Then validation of the cleaning process is a must on maintaining aseptic areas. Cleaning validation is also these days a big issue, that whenever any cleaning has been done, you have to check whether the cleaning is proper or not. And there's a big procedure and an SOP to check whether the cleaning was done properly or not. And after that, you come to know that whether any changes are required in the procedure of cleaning. Right? So this is how we learn from this case example. And I think this is the last one, I think. This is regarding imported dressings incident in UK. 33 out of 38 batches of Indian dressings were found to be contaminated. So it's a case of a export. Indian dressings from India, from one company, were exported to UK, and most of the batches were found to be contaminated. Contaminated was spore-forming cherubic bacilli and fungus. Investigation was carried out, and it was found the contamination was due to inadequate sterilization by the steam. Now, interesting point over here is that these surgical dressings are sterilized by autoclaving. And you know that when we do autoclaving, steam comes in contact with this material, and therefore some water vapor is absorbed. If you do not remove that water vapor from the covers of these surgical dressings, then they will remain wet for a long period of time, and then that will cause fungal growth. So this was seen that so the conclusion over here was that the post-sterilization contamination of fungus had to be taken place. So that is where one has to be careful, that drying has to be done after the cycle is over. So you stop the steam in the chamber, but you continue to have steam in the jacket. So the moist dressings become dry. Then only you stop the cycle and you remove the dressings. So that is how care has to be taken. If that is not done, some such issues can happen and your whole batch can become non-sterile. So these are a few cases. So we learned so many things, that what can go wrong and how it can go wrong. Now slowly we'll go into those cases also. So I'll not go into details of the formulation part. In the formulation, we have various components for manufacturing the product. Like in case of the major, if you see in an injection, you have the API, you have the vehicle, usually most popularly water for injection, if the drug is water soluble. Or if the drug is oil soluble, you may have some oils, where you may have arachis oil, sesame oil, or most popularly synthetic oils like ethyl oleate can be used. There can be co-solvents, buffers, preservatives, tonicity adjustment providers, wetting agents. So all these can add to the stability of the product. So that is the components, materials, methods and processes which are used, we'll discuss now, and the packaging components also. So these would totally come together and you'll get a product which you are developing. Okay. These are the when you're going to the aseptic processing. Primarily, we have two ways in which we can manufacture the injections. One is you have terminal sterilization, and second one is you have aseptic processing. In terminal sterilization, those products which are stable to heat, they can withstand autoclaving. That you can prepare by normal filling in a clean area, and then you can put the filled containers into the autoclaving for sterilization. That is safest. So after filling, you are sterilizing them. So even if there is any contamination inside, that is going to be killed. So the risk is less. But 70% of our APIs, they are thermolabile. And therefore, we do not have that option of going for terminal sterilization. So the rest, what option is left with us? We have to go for aseptic process. And that is the most risky because after filling the product, you are not going to sterilize it and it goes directly into the market after testing. If there is an issue, then your product can become contaminated during manufacture. So it's highly risky. And as I said, 70% of our products are prepared by aseptic processing. So these are some of the things which go into importance when we are working with aseptic processing. Like you have the traceability, what raw materials were used, what are the components used, what are the product, what are the change control, clean rooms where you are manufacturing the product, they are classified into different areas. Governing procedures have to be taken, traffic flows, which means movement of material, movement of people. So access control has to be there. Only those people who are required to go into that area should have access into that area. So unnecessary movement of man and material has to be avoided. Sanitization of all the areas should be done as per required norms. The time out of refrigeration, that is TOR, time out of refrigeration, which means if your product is highly sensitive to temperature, your API is also stored under refrigeration. And then after processing, you are going to create an injection which is also stored at a cool temperature. So the time out of refrigeration is to be checked. Otherwise, what happens if the cold chain is not maintained, the product will become unstable. Documentation, again, it's very important. You need a formal process of documentation. The documentation has to be controlled, it has to be consistent, and it has to be timely done. You cannot wait that, like we have these journals which you're writing, I have many students over here. So when we are writing the journals, we write it after 3-4 weeks. So at a time, we'll write those 3-4 practicals and then we'll submit it when the teacher will ask for it. That will not work. See, writing journals is one way in which you are being trained for documentation in the industry. Take it like that. That is how you will be able to do it in the industry, because in the industry you cannot prepare a document after the work has been done after a few days. No, as soon as the work is being done, you'll have to do the documentation concurrently or as the batch is over. So these are practices which we need to learn when we are in the institutions. Procedures are there again. The procedures are documented, controlled, followed exactly as written. So that's about the SOPs. Changeover, cleaning, validation, proof of training before and after. So this is how we are training people also. We are doing the validation also. And whenever changeover is being done from one product to another product, specific procedures have to be followed. Validation is a very important aspect. It needs a lot of detailed discussions because each and every process, each and every product has to be validated. So validation and calibration are very important, and we need specific another big time to discuss validation in detail. So we have rooms, equipments, processes, and the environment. These are to be validated so that the aseptic processing is done in a controlled environment which is validated. So the risk of contamination and product failure is not there. So this is a big chart in which all the things have been put together. So coming to the processing problems now, major and minor. Major is failure of sterility, particulate contamination, pyrogenicity, and content. That's the assay. Minor is fill volume, container closure, integrity, irregularity. These are some minor issues, and major issues which you may see in parenteral products. Now, this is how you see whenever there's a problem, there is a complaint, and sometimes you must have seen that product has to be recalled from the market. Because the product has been found to be of substandard quality, the product has to be recalled from the market. And that is a big issue for the company in two reasons. First thing, there is commercial loss, financial loss. Second thing is the reputation of the company is spoiled because the whole batch has failed, and if the company is a big company, their reputation is also spoiled. Therefore, there are various issues which arise from such recalls, that the product is in the market and it has been tested, it has found to be of substandard quality. So the FDCA or the Drug Regulatory Authority will issue a notice to the company that, see, this and this batch of this product is found to be of substandard quality because of this reason. Therefore, you have to take it back from the market and destroy it. So reasons for recall: there's a complaint by the customer. The product has gone to the customer and he sees that there's a problem in the product, he'll make a complaint. Detection of a GMP failure after release. The detection of a GMP failure can be by two cases. One is when there's an inspection. The authorities may find that there's a particular GMP failure where the SOPs have not been followed. Second thing is after release, the company itself finds that there was a problem. The company itself finds that there was a problem, that the batch has been released, it has gone into the market, and then afterwards the company finds that there's an issue, they will initiate a recall. Then there's a ongoing stability testing going on. Whenever a batch is manufactured, samples of each batch are retained up to the expiry date. Now, these batches which have been retained up to the expiry date, they are also sampled and checked for quality at periodic time intervals. And during that time interval, if any batch is not seen of proper quality or there's a problem which is seen in any batch, then the company itself will do an investigation, and they will, if it is found that the product has gone bad or decomposed, then they will initiate a recall that let us get this product back from the market because it has degraded. Then request by the national authorities, which means the regulatory authorities have done some testing and they said that this product is not complying with the requirements, so he has to call back. Results of an inspection, as I said. Known counterfeiting or tampering, which means there is adulteration which has been done, or tampering, which means people have played with the package. The drug is something else and the label is something else. In such cases also, the product has to be withdrawn from the market. We have seen recently in the newspaper, a lot of duplicate remdesivir injections are in the market. So this is also what happens, that, see, the container will have the label of the company. So first, when the product is caught, they will contact the company that, see, this product of your company is substandard. The company will then see the product, they say, this is not our product, this is the counterfeit product. Somebody has tampered with the product. A counterfeit product means somebody has prepared a product and they have filled something else inside and they have done it. A tampered product means the container is original, but somebody has removed some product and filled another product inside. That's called tampering. Counterfeit means everything is wrong, duplicate. Both these things can happen. In such cases also, if such products are confiscated from the market, they have to be recalled. Adverse reaction, sometimes it happens that the severe adverse reaction is reported when a particular injection is administered. In such cases also, this can be reported by the hospitals also, that when the hospitals are using injections or the doctors are administering injections, they find that there's a problem when a particular injection is being administered. They can do the reporting, and on the basis of that reports, the whole product can be recalled. So these are reasons of recalling. Sometimes what happens is that there's a big problem that after administering the product, people may die, which we have seen in the early examples. So these recalls are classified by the FDA and WHO also. So this is how the recalls are classified. Class 1 is recalls of for products which could cause serious injury or death. That if this product is administered, the patient is liable to die. In such cases, you have a class 1 recall, where within 24 hours you have to stop the distribution of this product. That's called class 1 recall. Class 2 is recalls for products which might cause serious injury or temporary illness. It may not be fatal, but it can cause serious injury. In such cases, you have class 2 recall. That's within, you can say, 48 to 72 hours. A class 3 recall is for minor problems which can cause injury or illness, but it is just a minor problem, like some labels are container closure problems are there or something like that. So in that case, class 3 recall is there. Sometimes what happens on a container, because of improper gum, the label is coming off. It's a very simple typical problem that on the injection there's a label. Now after storage up to 2-3 months, the label comes off from the injection vial or the ampule. So you do not know the identity of the container afterwards if the label comes off. So these are class 3 problems where the product is good, everything is good, but a minor problem of labeling is there. So that is how it, but it has to be withdrawn from market. So this is how the initiation is done. This is some of the latest information which maybe I collected yesterday only. This is of last week only, 4th of May. 2 cases were there. 3 cases were in March. This is from US FDA product failures withdrawal. You can see. You can visit the US FDA web page, and on the website there's a nice section on recalls or withdrawals. So you can see I just classified the injections from that. There's a section on injections. There's a section on drugs, section on biological, section on cosmetics, veterinary. So it's nicely you can get all this information. So the first example is there, which is shown that on 4th of May, this product of Hospira was recalled. It was 5% bupivacaine hydrochloride injection, USP 30 ml, and 1% lidocaine. Due to mislabeling, as I said, there's a problem of labeling, and because of that, the product has to be withdrawn. Another one was on 4th of May, sterile water for injection, USP 100 ml, single dose, glass flip top vial, due to visible particulates. So there's a problem of particulate matter, and because of that, the product had to be withdrawn. Then acyclovir injection, 50 milligram per ml, 10 ml injection, and 20 ml vials. Crystallization was seen. Yeah, this product was Zydus Pharmaceuticals. Now, this is they have their subsidiary industry. Zydus has a subsidiary in US, and this product was from there. And this was also recalled due to crystallization of the API. Again, a problem. Then lack of sterility, phenylephrine hydrochloride injection. It was found to be non-sterile when it was tested in the market. Sargent Pharmaceuticals. Enoxaparin sodium injection, packaging error resulting into an incorrect package. Apotex Corporation, again a very good company. You can see various reasons are there. You know, see mislabeling, particulate matter is there, crystallization, which means there's a formulation problem, there's a processing problem, sterility is not there, and packaging problem is there. You can see, so you know, various types of issues are there. This is all because of different problems during the manufacture of this parenteral product. Okay, so there's a small, I'll go into small bit of detail of sterilization, because this is one of the major issues where products are failing in sterilization. Another important fact is that at the institution level, none of the institutions would be having a tunnel sterilizer, which is one of the most popularly used sterilization machines in the industry for vials and ampules. No college has a tunnel sterilizer as far as I know. We always try to use a small hot air oven. So the conditions which are used in a tunnel sterilizer are very different from a hot air oven, and therefore there's a technological gap over here. So I'll try to just go through that and let's see. In case of moiety sterilization, what is listed is normally we use 15 psi, 121 degrees centigrade, 15 minutes as the normal standard for sterilization. I hope this everybody would be using and knowing. But the pharmacopoeias, all of the pharmacopoeias state this. Other combinations of time and temperature may be used, provided it has been satisfactorily demonstrated that the process chosen delivers an adequate level of lethality when operated routinely under established tolerances, which means the limit in which it is found to be lethal or able to kill the microorganisms. In establishing the sterilization cycle parameters, the F0 concept may be used. So, whenever you are going to use any other conditions for sterilization which is not 15 psi, 121 degrees centigrade and 15 minutes, in such cases, the recommendations are that you should monitor the cycle using F5. Now, normally in our institutions, we do not monitor any cycle or we do not learn how to calculate the value and to monitor any cycle on the basis of that. Whereas in the industry, many processes are such. Very common example I'll give you. All IV infusions packed in plastic bottles. All IV infusions packed in plastic bottles, which are usually 90% product you see in plastic bottles these days. All these are not amenable to 15 psi 121 degrees centigrade. Why? Because that plastic becomes soft at that temperature. So you are not able to use 121 degrees centigrade 15 psi 15 minutes. You are therefore compelled to use a lower temperature. You are forced to use a lower temperature because otherwise the bottles are becoming soft. Now, so you are deviating. You are deviating from the standard conditions. In such cases, see if you are using 110 degrees centigrade for sterilization. Okay, 110 degrees centigrade for autoclaving. It is not 115 or 121. So you will have to increase the exposure of the tank. That how much you will use. It has to be decided on the F value. Therefore, this is a major challenge which we should know, that how do you operate your autoclaves when you are having a product which cannot be sterilized at the standard sterilization cycle. So, in general, for aqueous preparations, a microbiologically validated steam sterilizing process that delivers an F value of not less than 8 to every container in the load is considered satisfactory. So, what is F value? F value is equivalent sterilizing time at 121 degrees centigrade for 15 minutes, right? So this is how we have to understand the application of F value when we are sterilizing an article which is not being sterilized at the standard sterilization cycle of 15 psi 121 degrees centigrade for 15 minutes. In such cases, this F value has to be used for monitoring the cycle. So this is where a gap is there. We need to learn this. Similarly, in case of dry sterilization, as I said, in the industry, we use tunnel sterilizers most popularly for continuous processing. None of these big industries are going to use a hot air oven. And another impact is that when you're using a hot air oven, like say 100, this is what is the old criteria which is listed in the books of sterilization or microbiology which you learn. 162 hours, 171 hours, 180 degrees, 30 minutes. Okay, but okay, your product will be sterile in this. But what about pyrogens? Will they be destroyed at this temperature? It's important because see, I tell you, this for all your water for injection which you're going to use, that's going to be sterilized, and maybe you're going to use this temperature for sterilization. If you use, then this is a dry sterilization. You are going to sterilize your vials and ampules which you're going to fill in dry sterilizers or hot air ovens or tunnel sterilizers popularly. When washing is done in automatic machines, some amount of water stays back in the vial or the ampule after washing. Now this water may contain pyrogens, and that water which remains in the container, when it is put into the sterilizer, if it's hot air oven, say 180 degrees centigrade for 30 minutes, we have sterilized it. So the vial becomes dry, it becomes sterile, but pyrogens are there inside. How do you remove them? There's no way. It goes into the product. It goes into the product. Now this is an issue, and therefore industry changed these parameters, and in the tunnel sterilizers they are using a temperature not less than 250 degrees centigrade. All radiant heat tunnel sterilizers, and they do not operate at a temperature below 250. Very high temperatures. Why? Because at that temperature, we have two benefits. Sterilization is also taking place. Depyrogenation is also taking place. So the products are safe, right? So that is why here also all the pharmacopoeias are saying that other combinations of time and temperature may be necessary for certain preparations, right? So that these are certain basics which we need to understand because we are deviating. We are deviating from the normal cycle, and therefore we have to validate our process, and we have to ensure that it is having the required sterility assurance level. It is giving you the required sterility assurance level, then it is acceptable by the authorities and it is safe. So that is something where a challenge is there, and we are not exposed to this in the institutions when we are learning this sterilization practices in our colleges. So as you know, this is about the D value, and you know that D value is one time required for 1 log reduction in the microorganisms. Okay. So here you can see over here the sterilization specifications and sterilization parameters are given over here. The F value is calculated by the temperature in the product and the time for which the product is held at that temperature. So these two things are important. When from this you can calculate the F value, and it can be done electronically also, and it can be displayed on the modern autoclaves. And you can get an value that now the F value is 5, 6, 7, 8. It is slowly increasing. You can reach the required F value, and then you can stop the sterilizer. So that is about a chart which is showing you how the specifications are there, machine settings are there, load configuration is there, which means the loading pattern in the autoclave. Let me stand over here. Loading pattern means if you do not load your particles properly inside the autoclave, then also there is a chance of failure. Though the parameters of temperature and pressure will be correct on the chart and the dial, the product will fail because it was not put inside the autoclave properly as required. So these are things which have to be taken care of: temperature setting, time setting, other pre-vacuum and other settings, containment of individual items, and loading pattern in the autoclave. These are things which have to be taken care of. Let's skip this. Okay. SIP is steam in place. That is, you are trying to sterilize and tank. Say, for example, you are making a tank. You're using a tank for preparing a batch. It is a 200-liter tank or a 500-liter tank, depending on the batch size. When the batch is prepared, you have to wash it so that the next batch can be prepared. How do you wash such a big tank? You cannot dismantle it and take it to the wash place because it's very huge. If you are preparing intravenous infusions like normal saline, dextrosaline, the batch size may be 5,000 liters, 10,000 liters. How do you wash that containers? It's a big challenge. In such cases, you have to wash the containers also, you have to wash all the pipelines also. So you have to clean them in place, and then you have to sterilize them in place wherever they are. So it's very challenging. So we have CIP, SIP systems. SIP means steam in place. The systems are sterilized in the place, and that is very complex. It's very complex because you cannot put a big tank into an autoclave. The tank has to be sterilized where it is. So you have to take a steam pipeline over there. You need a proper system over there where the steam is put inside the tank. Pressure is created. You hold it at 15 psi for 121 degrees centigrade, and then you stop it after the required time. It's pretty challenging. So this is where there can be some gaps and non-sterility can happen, right? So that is where we have this. So we need to learn about CIP, SIP systems, which is required, and this is very popularly used in the industry. But at the institute level, we do not focus on such learnings. I'll skip this. Okay, selecting the right agent for the sterilization of clean rooms. You know, clean rooms have to be maintained aseptic, and how to maintain them aseptic? There are various methods which are available over here. Normally, one of the most popular method is fumigation. And in fumigation, what do? You are using formaldehyde usually. So the formaldehyde is created in vapor form in the required concentration per cubic meter, and that kills all the microorganisms which will be present. So there are two ways of doing it. Formaldehyde usually either you have a vaporizer, so you are creating a vapor of formaldehyde, and it is circulated in the room in which has to be sterilized. So here we have four systems which can be used: manual spray and wipe techniques, which means you have tables and surfaces of machines which you want to sterilize, and you can use a spray or you can use a which is mop which is dipped in a disinfectant, or let's say it is 70% isopropyl alcohol or 70% ethanol or something like that, and or another disinfectant suitable, and it can be mopped and it will sterilize the surface. Okay, so it's a disinfectant which you can use. So you can use manual sprays, wipe techniques, nesting or fogging systems can be there, vapor systems can be there, and gaseous systems can be there. So these are things which you can use for maintaining the aseptic conditions of the clean room. Very important, as I said, because if the clean room is contaminated, then the whole product which is being manufactured inside will also be contaminated, right? So this is, and then you need to validate the cleaning also. As I said, cleaning validation has to be figured out whether your area is proper or not, and that is important. There are various procedures which are being used to ensure the environment inside the aseptic area is good. Okay. Manufacturing of high potency drugs using isolators. Isolators are completely closed units which are equipped with filters. These are HEPA filters which are there, and they are able to ensure preservation inside them, class 100, or also you can call it as ISO 5, and where the environment is also biodecontaminated in a reproducible manner and validated. Therefore, there are two things which happen over here. One thing is that there is no user intervention. The worker who is standing will not put his hand inside directly and touch the product because it is closed. We'll have a figure at the end to see the isolators. So there will be holes. The whole machine is covered on a conveyor. The product is passing. There's a HEPA filter on the top, so contamination does not enter from anywhere. And there are holes and there are gloves inside. We call them glove hands. So person can put his hand inside the glove and make some corrections if required. So that prevents any contamination from touching the or entering the manufacturing line. So these are known as isolators, and they are very effective for specifically for high potency drugs. Okay. Particulate matter is a big issue. Sources of particulate matter, we have materials, the raw material. The process can lead to particulate matter. Your equipment can lead to particulate matter. The manpower or people can shed particles, and that particles can go inside the product. It's very interesting. We as human beings, on the skin surface, every 3 days we lose one layer of skin. Every 3 days we lose one layer of skin. Where does that go? Are we able to see it? No, it's particulate matter. It goes into the air. So if people are working inside the aseptic area, and if their hands and feet and face are not covered, they will be continuously shedding particles. And this can enter into the product, it can contaminate the product. And therefore, when people are working inside the aseptic area, they have full gowns, headgear, overalls, booties, masks, everything is there. Why? Gloves, so that no part of the body is exposed, so no contamination would enter into the product. So particulate matter has to be controlled because these are the sources of particulate matter. And if particulate matter is present in the product, those containers are rejected. So this is about environmental monitoring. We have microbiological monitoring, we have particulate matter monitoring. In the case of microbiological monitoring, we have air samples, surface swabs, and personal swabs. What is done over here is air sample means you have a machine inside the aseptic area. In the machine, you place a ready-made sterile nutrient agar disc. A ready-made sterile nutrient agar disc is placed inside the machine. When you start the machine, air is sucked inside the machine. That air passes on the nutrient agar plate. for 1 hour you are keeping that machine on, and then you stop the machine, remove the agar plate, close it, and incubate. On incubation, if there is contamination, you find some colonies are growing, then there's a chart that depending on the area, this many number of colonies are acceptable. If you're finding more than that, there is a problem. That the area is not clean, there are more number of microbes in the area. Okay, so that air sampling. Surface swabs are possible. You are taking cotton balls, you are sterilizing those cotton balls in an autoclave, and then those cotton balls are then swabbed on surfaces of floor, machines, and then they are incubated in sterile nutrient broth. And if they find that there is turbidity which is being developing on incubation, they check that this swab was from which place. It was from the floor, which means the floor, though it is being disinfected, it is showing microbial growth or presence of microorganisms. So either the cleaning process is not good or the disinfectant which is being used is not effective. Then personal swabs are there, where you take the swab of sterile cotton and on the hands of the people who are wearing the gloves, you are putting those swabs and you are then incubating it. If you find contamination in that sample after incubation, what do we understand? Two things. Either the gloves are non-sterile, they have not been sterilized properly. You check the autoclave cycle, they were sterilized. Second thing is improper SOP. The people are not working properly when their hands with the gloves on, they are touching somewhere else, they are touching the face of the body and all that. So they are contaminating the gloves also. That is dangerous. So this is environmental monitoring by using microbiological samples. Then physical, where we have particulate matter, differential pressures, air changes, airflow patterns, cleanup time, filter integrity, temperature and relative humidity, and airflow. These are the physical parameters in an aseptic area which have to be monitored. I do not know how many of you might have the exposure of these parameters, how they are calculated in the industry, because you need to work in such areas, and then you are able to understand that. Differential pressures are required so that whenever any door is opened, the air from outside does not enter inside. So there is a differential pressure. The most critical operation is carried out under high pressure. So there's a lobby and there's a room of filling room. So the filling room is under positive pressure, lobby is under negative pressure. So if the door is opened, air from the lobby does not go into the filling room. Air from the filling room may go out into the lobby. As per the guidances, regulatory requirements, a differential pressure of not less than 15 pascals is required. This is the standard. So your pressure cascade has to be maintained like this. Then only your contamination levels will be controlled. Otherwise, there is all the chance that contamination may enter into a product because it's an aseptically filled product. So these are all risks which I have done. So air changes, it's very important. There's a regulation which says that if you're having a class 10,000 area, that the air changes should be not less than 30 air changes, 20 to 30 air changes per hour, which means the whole air in the room has to be changed 30 times in 1 hour. So you need to design an AHU for this purpose. We'll come to the design of the AHU also, and you will be able to understand. It's very easy. You know the length of the room, breadth of the room, and height of the room. So you know the volume of the room in cubic feet. You want to change that volume 30 times. So multiplied by 30, you have the total volume of air which has to be handled in 1 hour. You select a blower which is going to that CFM of air, cubic feet per minute of air, right? So blowers are available in CFM capacity, right? Or LPM, liters per minute. So do the calculation and select a blower which is giving you a higher efficiency than what is required, and you can design it and you can have it, and that will manage the air changes. Now, as per regulations, the number of air changes required is different in different areas. If it is a class 10,000 area, it may be 30. If it is a class 100,000 area, it may be 20, 10, 5, like that. So depend. If the cleanliness area is less, air changes required is less. If the clean area is high, critical area, air changes required is more. In our laminar flow benches, which we are using where the filling operation is taking place, that's a grade A area. You cannot place a a laminar flow bench in a uh, common area. Otherwise, you will not get the quality of air, right? So that is how the cascade of cleanliness has to be maintained. So that is the physical monitoring of the environment over here. So these are the parameters which are to be monitored when we are using aseptic areas. This has to be done regularly at time periods. Sanitation has to be done. There is a disinfection which is done as we have discussed all this. It has to be a documented procedure for cleaning and it has to be validated also. Monitoring of disinfectants and detergents has to be done to check any resistance has not developed. So that is also there. Clean containers stored for defined periods of time and sterilized before use when used in grade A or grade B areas. You cannot keep containers which have been sterilized also for more period of time. You'll have to re-sterilize them. That's not possible, not allowed also. So we discussed settle plates, volumetric air samples, surface sampling, swabs, and contact plates. Sampling methods should not contaminate the area. Now that's also important. When people from the QC lab, they will come and they will place the petri dishes at different locations in the aseptic area. On the machine they will place, on the floor they will place. They will check near the air which is coming from the AHU. There also they will place one. They will hold one petri dish. So at different locations they will place the petri dishes containing sterile agar, nutrient agar, and after 1 hour they'll take those samples and they will incubate the plates. And from that they will get the plate count. Okay, so one has to be careful that those plate counts are done properly and the proper procedure is followed. And this is the table which is given, like these are the requirements which have to be followed. The limits are given over here. Grade A, B, C, and D. These are the cleanliness levels which are there. Grade areas. These are also classified into by US Federal Standard 209D and 209E, where we are classifying them as in 209D we are saying in class 100, class 1000, class 10,000, class 100,000. In case of ISO, we have grades 4, 5, 6, 7, and 8. So these are how various ways in which the classification of clean areas is done. And these are the limits that I have given. You can see these limits and accordingly you can say whether the cleaning is proper or not, or the quality of the environment is proper or not in the aseptic area. Water is very important because we all know that water is one of the most popular vehicles which is used in the production of injections, right? Therefore, how is water manufactured for parenterals? Okay, so we have these processes: filtration, disinfection, reverse osmosis or deionization, distillation or ultrafiltration. So out of this, usually the last step is distillation. We have multi-column distillation skills. So before that you may have an RO, and before an RO you may have a DI, right? So these are, and before DI you have a filtration section which is there. So it's a water treatment plant which you will find in the farm industry. Those who have visited, you might be knowing about this. I'll show a small glimpse of what are the methods which are being used. This is a typical deionizer design where you have two columns. One is a cationic column, one is an anionic column, and any raw water which is coming from the water softener. So the water is coming from water softener. It is passing through the one column, then it is going into the another column. So anions and cations will be absorbed over the induced columns. Then it goes to a cartridge filter 5 micron, another cartridge filter 1 micron. It is ozonized, so there's an ozone generator. It is ozonized, and then it is heated over here to the required temperature, and it is again going over here for recycle. So this is the recycle loop. So that, see, water must be kept circulating. Why? If it is not kept circulating, then there are chances that pyrogens will develop. So to prevent the development of pyrogens, see, pyrogens once developed, it's very difficult to remove from water. You cannot remove pyrogens from water if they are developed. So the option is you have to take care that pyrogens don't develop. As you all know, pyrogens are metabolite products of gram-negative bacteria. They are filterable. They are non-volatile, heat stable, and when injected in the human body, they cause a fever response. and it may cause shivers, and if currents are high, patient can also die. So we knowing these properties, we have to manage that the pathogens don't come into our water, because water is going to be used as a vehicle in the manufacture. Therefore, water has to be kept circulating. So there's always a circulating loop where the water is kept under circulation. It is not stagnant. Dead spots or stagnant spots allow breeding of microorganisms. Okay, so it has to be maintained around 70 to 80 degrees and kept in constant circulation. So that is how this is the system for deionization. This is not sterilization, mind you. This is just deionization. You're removing the ions, positive and negative. That's anions and cations are removed by this cup. Then this goes into the RO. This is a schematic diagram for the reverse osmosis, where you are having a high pressure over here and purified water comes out over here. So this is the mechanism which is being used, and you must have seen that at many in the house also you install RO systems for water. There's a lot of waste water coming out. So reject water is coming out over here. You can see over here, and only some amount of purified water comes out, which is not containing any anions, cations, or any such molecules. They are, because they will not pass through the semi-permeable membrane, right? So reverse osmosis is used, and this is the industrial design. You can see. This is the design of a typical industrial RO, reverse osmosis plant, and you can see over here, water is coming from the DM plant from the earlier stage. It is coming over here. First is buffer tank. From a pump, it is going to a filter cartridge. Again through a pump, this is the RO cartridge. First stage RO cartridge. Then it is going to the second stage RO cartridge, like that, and it is recirculated, and if required, it is taken out for manufacturing. Here you can see again, this is a heater. So it is taken care of, that it is kept heated. It is also kept under circulation. This is written over here. Water returns to the first stage buffer tank, so it is going back over here. So that recirculation is being done to maintain the water under pathogen-free conditions. So this is interesting. You do not see something like this in any institution, right? So the industries always have this type of plant, which is commonly used. So we should need to know that what can go wrong if water is not properly manufactured or properly made, water for injection, right? So these are different stages in which your water for injection is being manufactured. This is the second stage. And then you have the third stage in which you have multi-column distillation still. This is the multi-column distillation still, where the water which was coming from the RO unit is fed into this, and you have steam coming inside. Two things are there, and only cooling water in the condenser is there. These are three things which are going into the multi-column distillation unit, and you get water for injection coming out. Okay, so it is also kept under constant recirculation, and it's a very efficient system. This is the most commonly used equipment for manufacturing water for injection, as you all must be knowing. A very nice design, and it is very efficient, very efficient, right? So the cost of production of water for injection should be maintained as low as possible, because this is the vehicle, right? So this water should not be costly. So these designs are very well managed, so that the cost of producing water for injection is less. So what goes in? Steam, water, from which water is vaporized, and that vaporized water is then condensed in the condenser, and you're going to get water for injection, right? So this is it's a complicated system. You need to understand it very nicely. When you go to the industry, when you go for training, you can see the working of this, and you learn how it operates and what are the problems which can happen over here. That is interesting, right? So try to learn about the multi-column distillation units and how the problems can be there in the water which is being coming out from this multi-column distillation units. This is one of the major sources of making water for injection. Okay. So what are factors contributing to the quality of our product? So all these have been put together over here now. You can see personnel, very important. As I said, procedures, equipments, premises, environment, packaging materials, the raw materials, and the validated process. All put together, this is the quality chain. Right? Each and every part is having its own importance. If one of them is not proper, then the quality chain breaks down. Okay. So this is what I will listed over here. Light manufacturing environment, if we go to see, it is critical. Light, temperature, humidity, air movement, microbial contamination, particulate contamination, as we have discussed. These are things which are important. We know how the material this can be controlled. For controlling temperature, humidity, air movement, for this and microbial particles. For all this except light, the rest of the things are controlled by one equipment, or which is known as HVAC system: heating, ventilation, and air conditioning. So the HVAC system controls temperature, humidity, the air movement, microbial contamination, and particulate contamination. All this is controlled by the HVAC unit, our system. So very nice, complicated system. I do not know how many institutions have an HVAC system. A few lucky institutions where the aseptic areas were good, the students have an opportunity to visit and see that AHU in working condition and understand how each and every part of the AHU is working, because that's very important. As per regulations in the industry, each and every department needs an AHU or a HVAC system. Even in the tablet department, capsule department, injection department, everywhere, each section needs a separate AHU. So these AHUs or HVAC systems become very important because they are critical for maintaining the required environment inside. Okay, we'll see one design also. We'll see a design of the AHU afterwards, and you'll be able to get an idea about the complication of that design. Okay? Contaminants are important because if the environment is not good, you can have contaminants in the product. Okay, so contaminants are products or substances other than the product which is manufactured. Foreign material, particulate matter, microorganisms, endotoxins, which are dead or degraded microorganisms. They are also contaminants. Cross-contamination is a particular case of contamination. Cross-contamination means products of substances other than the product manufactured they are entering. Okay. So, what is cross-contamination? Contamination of a starting material, intermediate product, or finished product with another starting material or product during production. So, if you have two products being manufactured side by side, and if the cleanliness levels are not being maintained, then one traces of one product are entering into the other product. Or the same equipment was used for manufacturing one product, and that same equipment was used for manufacturing another product. It was not cleaned properly. Then the traces of the earlier product will be seen as contaminants in the next product. This is known as cross-contamination. So one has to be very careful that cross-contamination does not take place. Right? So what is the cause or reason for cross-contamination? We have poorly designed air handling systems and dust extraction systems, poorly operated and maintained air handling systems. You have inadequate procedures for personnel and equipment, and insufficiently cleaned equipment. That's what I said, that if the cleaning is not proper, contamination will definitely take place. So this is about cross-contamination. I'll skip this. Okay, here we are. The levels of protection, therefore, which are required, because one has to be careful that you are going to have the right type of area which you're going to use for different processes in the parenteral department. So WHO is classifying areas as A, B, C, and D class. European Commission, PICS, TGA, they are also using the same classification ABCD. US FDA is using critical and controlled environments. ISPE is using level 1, 2, 3 or clean room class. So companies they have an option of using any of these classifications. So this is how it is. What process? Say washing of containers, class D or grade D area. Preparation of solution for terminal installation, grade C. Preparation of solutions for aseptic filling, A, B or C class. Deparogenation, A. Filling for terminal installation, C. But filling for aseptic process, A. See, why this is different? When you are filling a container for terminal sterilization, your container is then going to be put for autoclaving. So even if there's some risk of contamination, that is going to be killed when that container is placed into the autoclave. Whereas in case of an aseptic process, You're not going to go for terminal sterilization. So whatever you are going to fill has to be filled in a very clean environment without any risk of contamination. So that is where one has to be careful. So it is done in grade A. Okay. So that is how we are saying about the levels of protection. Based on the clean room class requirements, various levels of protection have to be created. Correlation between process operations and clean room classes. The type of operation permitted each level of protection. The definition of the clean room class. Parameters, building materials, room requirements, and HVAC system. What this means is that if you want a class 100 area, then you have to be careful that how to design a class 100 area. Because when you are designing a clean room, the design also incorporates certain features like what will be the floor, what will be the ceiling, what will be the walls, what will be the paint, what will be the material of the flooring, how much amount of air, from which side of the room you will introduce the air, how many risers will be there, the exhaust air will go from which place in the room. All these are technical things which are to be designed so that your area is working efficiently. Otherwise, when you validate the area, it may fail. Right? So depending on the level of protection required, the whole area design and the processes will be designed like that. Requirements on entry conditions for personnel and material. The change procedures also will be arranged according to the level of protection required. So you'll have change rooms, you'll have crossover benches, you will have pressure cascades to maintain the level of protection. I was talking about HVAC or the air handling units, which are very critical for maintaining the proper environment. So this is the design of an AHU, where the AHU, you can see central air handling unit, this will be outside. This is outside the septic area. Fresh air is coming into this AHU. The AHU is filtering the air, it is reducing the temperature to the required temperature, it is removing moisture also, so dehumidification takes place, and it is filtered, and then it is going into the production room. So this is the room in which that treated, filtered, sterile air is entering. This is the terminal HEPA filter in the production room, from where sterile HEPA filtered air comes into the room. And from there, this is the return air duct. It is going out. Exhaust air. This is the damper. This is the silencer, filter, and it goes out. This is a typical system where 100% exhaust is there. No recirculation is shown over here. Mind you, there are two types of AHUs. One in which there is recirculation of the treated air. Second one is where if you are using that area for manufacturing of beta-lactam antibiotics, then or cancer drugs, then it is a mandatory requirement that 100% exhaust should be there. Air cannot be recirculated. So in such cases, this type of system will be there, where fresh air is coming inside. There's a louvers over there. This is the air valve to control the amount of air coming inside. There's a blower, you have the filters, then the cooling coil, then the humidity control or the humidity generate air vapor generator or the vapor removal, and then the pre-filter, final filter over here, and then HEPA filter over here. And this is the regulator of the amount of air which is going inside. Right? So this is how this unit operates. When 100% exhaust is there, there is no recirculation of air. There is another system where you can see over here. This is also, I'll show you over here, where you can have a recirculation of air, and if you want, you can have 100% exhaust also. So the components are also shown over here. I'll show it to you. Very few, I think very few, as I tell you, very few institutions have such AHUs in their campus, in the aseptic areas. So it is interesting to learn if you have come across some such AHUs. This is the entry of the outside air. There's a louver which prevents insects or birds and flies. Then there's a valve which controls the amount of air going inside. This is a pre-filter. Then you have this heating coil over here. You have the separator, the cooling coil, water droplet separator, which means when cooling coil is there, you know what happens when there's a water which drops out, like even when you're having AC, water comes out because there is condensation of water vapor. This water vapor has to be collected, otherwise that water vapor droplets will go into the clean room, because they will be very fine droplets. You have to strain them, you have to remove those water droplets, so that separator is over here for that purpose. Then if required, if the humidity is less, then you have to add humidity. If the humidity is more, you have to remove humidity. So then there's a secondary filter over here. The air from that is coming out. So this is known as your plenum. This part is known as your plenum, and this will be seen as a nice box outside in which all these components are put together. That is going to the control damper, which controls the amount of air going ahead, and the heater over here if required, or cool. In certain countries, like say European countries where the temperature is very low, and you have to bring the temperature of air to from 22 to 26 degrees centigrade, then you may need a heater. In countries like India, we never would require heater. We'll need refrigeration systems where we have to reduce the temperature from 40 or from 30 to 22. So depending on the type of environment in the country, the components of this AHU will change. Okay. So air is coming from the top. So the terminal HEPA filter is there. So you have a nice sterile filtered air. As you all know, the HEPA filter has an efficiency of 99.97% to remove particles of 0.3 micron larger. Right? So you get sterile air into the room. This is a production room. From there, that air goes out. Again, there's a filter, fan, rate controller, silencer, and exhaust regular. But normally, if you are producing normal injectables, you do not throw out this air. It is costly. It is all treated air. So you can see on the bottom there's a recirculated air. This air which is coming out from the production room again goes back over here into the plenum, and it is again filtered and treated and it goes back into the production room. So this is how you may have the recirculation being done. When you are doing fumigation, this is important. When you are doing fumigation of the aseptic air in this production room, then after fumigation is done, all the air has to be removed. You cannot recirculate it. So you close this valve, open this valve, and everything will go out. After that, when all the vapor of formaldehyde has been removed, you close the exhaust and open the recirculation. So that is how these systems are operated, and one has to be very careful that these systems are maintained properly. Otherwise, if the environment of the aseptic area is not correct, product will be contaminated. It will become non-sterile. So that's why this is the importance of the AHU or the HVAC system. Components are shown over there. I have listed all the requirements also. As you see over here, this is how the manufacturing processes take place. Everything is closed, shielded. On the top, you will see these are lamellar flow units. So the filling operation which is being done, this filling operation has to be done under class 100. Class 100 is achieved by a laminar flow bench. So you have the laminar flow on the top. This is a vertical laminar flow. So the air which is coming from the top is sterile, and all these operations are carried out under sterile conditions, and no person is touching. You can see over here there's a nice glass partition over there, so the operators are outside seeing the process taking place. They don't touch it, so intervention is very less and the contamination is controlled. Okay, so this is how under aseptic condition you can see the way in which all the gowning is done. You see, so there's no part of the body which is exposed, so no contamination will come from the operator into the product. Therefore, SOPs, gowning procedures, working the way in which you work, everything has to be, the training of people has to be done so that they are able to work efficiently. Then there's another technology which is known as BFS or FFS technology, that is blow fill seal or form fill seal technology. This is very popular because most of the products, IV products, except SVPs, they're all in this method, BFS. The interesting part is it's a very big market, and therefore we need to understand very nicely about it. Unfortunately, no institute has a BFS technology in the college. So students are not able to learn and work on such machines. Why? Because these machines are very costly. You do not have small laboratory prototypes of these machines available. only when you go to the industry, you see the working of these machines. And you don't know, you're not trained on this technology, so there's a big technical gap. So this is an area again where we need to work on and make the students understand how this BFS or the FFS technology is working. Simply in the books, if you see, we have a polymer. The Romelag is one of the most pioneering companies. It's a German company. It's the most pioneering company for for machines for FFS technology. Okay. So interestingly, also to note that in India, Core Parenterals was the first company which started manufacturing water of injection, normal saline, dextrosaline in plastic bottles. Earlier, this used to come only in glass. So this was in the late 80s, maybe around 80. After 85, the company was established in near Chatral in Gujarat, and they were the first ones to start using plastic bottles for filling IV fluids. It was very interesting that from LM College of Pharmacy, where say in 1994, 93, 94, we used to go to train their people in core parenterals. Very interesting, I tell you. Mr. Sunil Handa was the CEO, and we used to go with Mr. Sunil Handa to core parenterals, and we used to have training sessions over there, 15 days, to train their fresh recruited people. When they were recruiting people, we used to give them a crash course on parenterals and everything required for the parental whole setup for 15 days, and then they were inducted. So it was very interesting to get associated with the industry for such training programs where we used to go and train the people over there. So it's very simple. The plastic granules are fed into the machine, and it comes out in a melted tube form. So it's called over here. These are dyes. As then the dyes come together, air is blown, sterile air, so that plastic takes the shape of a bottle, and the dyes are having chilled water circulating in it. So as soon as the air is blown, the plastic comes in contact with the dye, it becomes cool, it becomes rigid. And then there is another tube which fills the product inside. After the product is filled inside, the tube goes out from the top. The upper portion is sealed with heat and the container comes out. It's a simple process of blow, fill, and seal, right? So these are very automated machines, wonderful. Very less risk of contamination because everything is done inside one machine. So this technology is a very proven technology, and it's very interesting to learn more about this technology. Unfortunately, we don't have small laboratory scale machines available, so there's a technological gap. But I would request, please learn more into detail about the engineering part of this. Okay, so these are modern production lines. If you see, where there's an unscrambler where you have the vials or empty vials coming over here, and then they're being filled. The rubber stoppering is done, aluminum seal is being done over here, and the product goes out. And at each place, this is on the top, you have a vertical laminar flow bench. These are the gauges, these round ones you can see, they are pressure gauges. They are showing the pressure of the air which is coming out from the laminar flow bench. So, the pressure cascade inside. They are getting class 100 environment inside, and the filling of the vials is done over here. Okay, the vials are being filled on a continuous manufacturing line. At each place you see there is a nice touch screen controller. Okay, so you can control the process by a nice touch screen. It's a computerized controlled process. So this is what you will see in all modern injection or parenteral manufacturing companies. Totally automated equipments are there. Okay. This is what I was talking about, isolators. These are, gloves. They are showing out. You can put your hand and this glove will go inside. You don't put your hand inside, you put your hand inside this glove, and then if you want to do any modulation, it can be modulated. So these are isolators, and these are the places from which a worker can put his hand inside the glove, and then this glove, and then you can handle if there's any problem inside, you can handle it. So these isolators are very popular, as we have seen an isolator earlier in another example also. But I was trying to show the glove holes. These holes are having a sort of like this glove, long glove. So the hand can go inside and your hand is protected by the glove. So the glove will touch the material, then your hand will not touch the material. So this is how isolators are there. Wonderful technology which is being currently used. As I was talking about CIP technology, SIP technology, as I said, it is for the cleaning purpose and sterilization purpose. So we discussed about this earlier. We'll skip that. There's a This is the design of a CIP SIP system. We don't have much time to discuss it, but what we are trying to say is that the component or the system or the uh, uh, tank which we want to clean. This is the CIP supply. The water from here goes into the cleaning tank. From the cleaning tank, the wash water comes back. It is going into this and it is retreated. Detergent is there, recovery tank is there, rinse tank is there. So there is a cycle. First you're washing with normal water, then you're washing with detergent, they're washing with again water. All that is cycle is designed, and accordingly these pumps are operating, these valves are operating, and it goes into the washing system. And the machines, the pipelines, everything is washed. Okay, so this is a multi-tank schematic design of a clean in place system, CIP. All parental machines of large size, all parental plants of large size will be having this CIP SIP systems mandatorily. Last we come to the automation. All these things, as I said, all these are complicated and automated systems. So a lot of automation comes in. So we have SCADA systems which are responsible for automation. Your touch screen industrial computer systems on which you can, instead of having switches, you have nice touch screen computerized vision available. So we can set all the parameters, you can read all the parameters, and we are connected with a server. You can get all the printouts on the server also. Programmable recipes are there for the processes where you can feed a recipe. Recipe means process parameters. If you are doing sterilization, then you can feed the recipe that for 5 minutes this will happen. The rubber stoppers will be loaded into the machine. For loading the rubber stoppers, you may have a vacuum conveying system. So for the vacuum pump has to be activated. After the vacuum pump is activated for 1 minute, vacuum pump should stop. Then the container in which the rubber stoppers have gone should rotate for 1 minute. Then water should be added into it. After that, it should be rotated again. Then you have a detergent which is added into it. Again, it is rotating. So, let's say, for example, you are having a automatic uh, clothes washing machine. Where you have machine one, kapda dhowanu. Similarly, like there's a cycle. So that cycle can be programmed for how much time you want each step. So that's a program we call a recipe. Each equipment has a recipe. You can program it accordingly. The machine will run. Proximity switches are important. That when all the processes are going in line, so we have to take care that each process is controlled by the next process and the earlier process. So proximity switches are used to control the on and off, for switching on and off of the two processes which are around it. Let's say if you are doing rubber stoppering, rubber stopper machine is having a problem, then the vials will be collected on the conveyor. So the filling machine has to stop. Otherwise, everything will be jammed. So there is a proximity switch on the conveyor that if the vials collect beyond this level, the filling machine will stop. If the filling machine stops, then the turntable on which the vials are coming from the sterilizer, turn sterilizer, it gets a signal. So the turn sterilizer will stop. And the turn sterilizer stops, it will give an indication to the washing machine. So the washing machine stops. So all these processes are controlled by sensors. And for such, proximity switches are one of the most important sensors which are used over there. So a lot of automation goes in. All machines are semi or fully automated. Vacuum conveying is done to a great extent to prevent personal intervention. Like as I said, if you want to put your granules into a hopper, you can have a vacuum conveying system. You don't need to put it by hand, by a manual person. Similarly, if you want to load your rubber stoppers into the autoclave sterilizer, there are special machines for sterilizing or washing and sterilization of rubber stoppers. Then you have vacuum conveying systems over there. So these are very popular. So these are all used for automation. So there's a lot of automation which is used in the industry, which you may not come across anywhere in your academics. So this is a big gap. You need to really look into how these automatic processes are being used and they are controlled. Okay. So last one I come to is documentation. We have finished. Our time is over. I think we have only 2 minutes now. This is a major challenge where students are not exposed to this, and because of that, they fail miserably when they go to the industry. They don't know how to write the reports. So they are not exposed to these reports in the institutions when they are learning as students. So I would request all the faculty members who are present over here, please train your students to try to fill the BMRs. That's the batch manufacturing record, or the batch production record, or the validation reports, or if media fill is being done, then the reports of media fill have to be filled. All these types of reports, we should show them the blank forms and then tell them what information has to be filled inside. Because when the student, as a fresher, he goes to the industry, he doesn't know what he has to fill. Each form is having a lot of information which has to be filled. He doesn't know any form. He has not seen any form. That's a big challenge. Therefore, I would request all the students also present over here that before you go, please learn how to fill a BMR, a BPR, validation reports. And see, this will help you because when you know all this, you are more confident. The industry also knows that you know all these things. So your progress is faster. So these are, major gaps which I have tried to address over here in this lecture, connecting these gaps with some theoretical points and trying to explain how things are happening in the industry. I'll just skip this. So these are the regulatory guidances which are available. You can refer USFDA. There are two sections, CDR and CBR. D is for drugs, B is for biologics. EMEA is for the European Union, European Commission. We have EMEA website. WHO is for the World Health Organization. It also is giving so many instructions and so many guidelines for manufacturing of quality and safety. ISP is International Society of Pharmaceutical Engineers. This is also giving guidances specifically for parenterals. PICS is the Pharmaceutical Inspection Committee Convention, and PICS is the Pharmaceutical Inspection Cooperation Scheme. Again, focusing more on parenterals also. And above all of this, you have ICH guidelines, which are also important for us. So with these, we have regulatory guidances available over here, which you can refer. In India, we have our Drugs and Cosmetics Act, and that also has all the requirements which have been listed for the manufacturing of safe, effective, and stable medicines. Right? So these are the technical WHO guidelines. You can refer. They are freely available on the WHO website. So I will conclude my talk over here that processing of parenteral products poses a real-time challenges at different stages, not just one. There at each point you have so many challenges which are there. Therefore, in-depth interpretation of the regulatory guidelines would be of immense help in taking the right decisions. With intervention of newer technology, the need for training and technical skill upgradation becomes vital. So again, training and skill upgradation, right? So these are important for us. So when we go to the industry as freshers from the institute, the more exposure we have about this, the more we get ready ourselves, the more acceptable we are. The industry does not need to spend more time to train us, and we are therefore much better placed. And obviously, salaries would be higher if you know the right things in advance. So with this, I conclude my talk. It's a very big area. I will just try to touch upon certain main aspects, and I hope a lot of questions will be raised in your mind. If any questions are there, I'll be happy to answer them.
Thank you so much, sir. Very nice and useful information regarding the bridging the gap between academic and industries. May all the students getting useful knowledge for this or upcoming future. Sir, now some question arise from some faculties and students.
Yes.
Sir, one question from attendee. how to fill the gap and which type of practicals we have to design in parental section.
Okay. So I would like to say over here is that once you make a list of the knowledge which the student needs in such cases, then there are two ways in which you can do it. One is you can have field trips. You take the students to an industry with a particular focus of teaching them a particular process. Like say today we are going to learn only parenteral processing, the filling, sterilization and filling. Take the students to an industry and show them only that part, full day. Then they will see each and everything. Then certain evaluation parameters, if you want to do, we can do it in the institution. You can show them videos. Videos are very nice videos are available on YouTube. You can give exercises to the students that see these are 5 videos which you have to see and come, and then we'll have a question answer session. So the students will see those videos, they'll have some queries in their minds, and then when you have a discussion, the learning is much better. So I would recommend two things. One is you wherever possible, if the facility is not there in the college, then we can do two things. One thing is we can take them for field trips, and second thing is we can have video presentations of actually the videos of each process taking place from YouTube and have interactive sessions. I think that will be very useful to the students.
Yes, thank you, sir. Second question is what is the importance of F value in the sterilization cycle development?
Right. As I said that normally, and see, F values or autoclaving only. It is for moist sterilization. It is not for dry sterilization. Clearly, I'll let you know. Second thing is in moist sterilization, we are normal sterilization cycle is 15 psi, 121 degrees centigrade, 15 minutes of saturated steam. Saturated steam, mind well, into inverted commas, because that's very important. If it's not saturated, sterilization will not be there. So when we are deviating from these conditions of 15 psi and 121 degrees centigrade, in such cases, F value has to be taken into account. Your cycle has to be monitored by F value. And it has been said that not less than 8 F value should be there. Then the process is considered to be satisfactory. So what F value indicates? It indicates equivalent sterilizing time. And F value indicates equivalent sterilizing time where a product is sterilized at a condition other than the standard conditions. So if you're getting an F value of 10, it means, say you are doing a process at 110 degrees centigrade, you're not doing it at 121 degrees centigrade, and you're monitoring the cycle by F value, then if you're getting an F value of 10, it means that if this cycle was run at 121 degrees centigrade, it was equivalent to 10 minutes.
Is there any possibility, sir, to prepare herbal parenterals?
Yes, absolutely, no issues. What you have to do is, see, it's a question of formulation, right? Preparing a product, one has to be very careful that when you're formulating a product, any herbal, or it may be an API or it may be a herbal. Point of interest is that whether any degradation is taking place or not, and whether the product is still having its efficacy. So the pre-formulation characteristics and all that have to be done properly when the stability and everything is being checked. If that is fine enough, we can prepare herbal parenterals also, not an issue. Only thing is whether you are going to market it as a phytopharmaceutical or you are going to market it as an Ayurvedic proprietary medicine. That's the question.
Yes, sir.
Because
Sir, next question is how can we determine D value and Q10 value?
okay, see, these are very simple theoretical values. D value means it is the time interval required for 1 log reduction, which means if you have 100 microorganisms, from 100 it becomes 10. The time required for reducing 100 from 100 to 10 is 1 log reduction. From 1000 to get it to 100, from 100 to get to 10, that's one log reduction. So we are measuring, what we do is, we can do one thing, you take a suspension of microorganisms, you take a suspension of microorganisms, do the counting, you do the viable count of the microorganisms, so you know the number of microorganisms present in the sample. Put that sample into a specific temperature environment, 60 degrees or 70 degrees or 80 degrees, and then at particular time intervals, you take out the sample and do the count again. So you'll have a graph.
Yes.
You'll have a graph on the y-axis. You will have total number of surviving organisms. On the x-axis, you will have time. So
Yes.
you prepare that graph. So it will be a reverse line. It goes down, right? And then, as it has been shown, from the y-axis, you can go from 1000, come down. From the y-axis, go to 10 and come down and see the time required for 1 log reduction. You'll get the d value. You will get the d value at that specific temperature for that specific organism. This is how you can find out d value. And similarly, you can find Q10 value. Same way.
Yes. Thank you, sir. One more question is why there is difference permeated solid content in water for injection and sterile water for injection? Sir, can I repeat the questions?
I can see it over here. No issues.
Yes.
See, water of injection and sterile water of injection. The difference is very minimal, I'll tell you again. But what is important over here is that when you're preparing sterile water of injection is prepared by multi-column distillation. Mind well. See, there's a slight difference over here. Water of injection, quality water you can get even from RO. But sterile water of injection, you are always preparing by multi-column distillation. So the solid content which you will get in a sterile water of injection will prepared by multi-column distillation will be very less or minimal than that water of injection prepared by RO or by any other method. Therefore, this difference is there.
Yes, sir. Thank you, sir. Next question is, in the books, it is given that FFS technique take a 6 seconds to produce a 1 container. It is so fast in an industry.
Absolutely, it is very fast. So I'll tell you, the dyes, they are 6 or 8 or 10 in number in one shot. So at the same time, when the dye comes together and comes out, 10 units are produced at the same time, or 4 units or 6 units are produced at the same time. So it's, the dye comes together, water, the product is filled inside, air is pushed, product is filled, and it opens out. Again, the product falls, again, the dye comes back again. It's pretty fast, and therefore you can see, if you can go to the website of Weiler, or you can go to the website of Romelag, and you can see the machines, specifications are given over there. All the productivity is also given that in 1 hour, how many units you can produce. It's very fast, very high capacity. True, it's very true.
Sir, last question is, what is SAL level? What are the method for the calculation of D value?
Okay, D value we have already discussed. SAL is very interesting. I did not pay much time on that. It's very important. SAL means sterility assurance level. Sterility assurance level means that the confidence at which you can say that, or the probability of having a non-sterile unit, an SAL of 10⁶ is achievable in moist sterilization, right? What this means is that 10⁶ means 1 in 1 million. 10⁶ is 1 million, okay? Which says that not more than 1 container in 1 million units should be non-sterile. This is what SAL means to us. A sterility assurance level of 10⁶ means less than 1 unit in 1 million should be non-sterile. So that's a probability. There is another term which is known as PNSU, probability of a non-sterile unit, PNSU. It's very interesting. And these are, see, in case of dried sterilization, you can go to 10⁻12 also, because dried sterilization processes are very strong. You are using heat over there. Whereas in case of moist sterilization, a sterility assurance level of 10⁶ is achievable. And that is what is the industrial standard, that you need to have an SAL of 10⁶, which means out of 1 million vials or 10 lakh vials, only probability of 1 or less than 1 non-sterile vial should be. That's what SAL means.
Thank you so much, sir. Now I hand over to Dr. Kaushik B. Kannada, sir. Sir is a professor of Department of Pharmacognosy at Saraswati Institute for vote of thanks.
Thank you, Dr. Jimmy, for giving me an opportunity. Namaste and good evening all. Dr. Praful Bharadia, sir. Today is our guest as well as the speaker for this webinar. Sir is my teacher. I studied under the guidance of the sir for more than 4 years, nearly 4 years in BFA. And sir already given the guidance about the engineering drawing, then unit operations, microbiology, and technology. Sir, same thing I am feeling now as a student of the BFAM when I was in the BFAM. Still the feeling will be the same. So thank you, sir. Guest, other principal, faculty members, and students across India who joined us. It is my privilege to pass on the vote of thanks on this event or webinar. I would like to express our sincere thanks to Bharadia sir. Once again, he is my teacher, giving an excellent coverage to the challenges between the in parental processing, bridging the gap between academia as well as the industry. Sir has given everything about the parental product starting means from A to Z, from starting from the why parental products are required, and it's nowadays in a corona pandemic we see that is the remdesivir injections. Now it is a layman word. Generally એવું થાય કે રેમડેસિવિર છે કે પછી બીજી જે ટોસિલિઝુમેબ છે કે એવા બધા જે ઇન્જેક્શન છે જનરલી ફાર્માસિસ્ટ અથવા તો ડોક્ટર્સને જાણીતા હોય, પણ નાવ લેમેન પીપલના મોઢા ઉપર જીભ પર આ નામ હવે ફરવા મળ્યા છે. So this is the need of the parental products. It is like in a case of the emergency, then various case studies which is related to the ignorance or SOP or that is failure of the components, etc. Sir has given the CGMP requirement, processing problems, complaint and recalls, sterilization technique with specification, sterilizing agent, contaminants which are frequently occurring for the sterilized product, and RO system. Even sir has given the diagram for the various AHU. It is the very good guideline or which is guidance to the students. If they want to prepare any model for the college college work, then they can produce this type of the AHU also. So thank you very much, sir, for giving us this very good guidance about all the things of the parental products. I would like to also thank our President, Dr. K.G. Patel, sir, Principal Dr. S.K. sir, and Ankit sir for giving us opportunity to organizing this type of the webinars for the benefit of the faculties as well as the students across the India. Once again, sir, I am very thankful to you for giving such a huge guidance for the parental products. Thank you very much, sir.
Thank you so much, Kaushik bhai. In fact, I tell you, the topic is really vast. I tell you, there was one line which was written in the last slide, that was media fields. There can be a 2-hour discussion only on media fields, because that's required when any new facility is being set up. You have to carry out media fields and you have to validate the area whether it is proper or not. So each and every line, which we are trying to say over here needs a lot of discussion. But because of the time constraint, I try to put something inside so that it can be an eye opener. Later on, if required, the students can go into these points into detail and they can learn more. So that was the intention over here. And Ankit bhai, I'll tell you, when I was preparing that slide of automation, I remembered you yesterday night.
Yes, sir. Thank you.
Because I remember I still have that thesis of yours with me, soft copy of automation. It's a wonderful work which you had done at that time in P-Farm level. But I still sincerely, I still think that 2 hours is not sufficient for this discussion. I just try to highlight a few things. It can be taken up with interest, and you can still work on it further with the teams. You can make teams, and each team of students can work on some particular problem, and then it can be group discussions. You learn best by case studies and group discussions. So wherever this opportunity exists, I would request you can have group discussions. That will really help. So I thank Ankit bhai again, standing by Ankit bhai, the management, for giving me an opportunity to come and talk to you all. Thank you so much.
Yes, sir. So one more thing I want to tell you, that you given the suggestion that we can give students to DMR and BPR.
Very
formats.
important. Very
Yes.
important. I do
So,
it.
yes,
I do it every time.
yes. So if we give, then student might be know about the details of formats.
Very true.
That is very good suggestion, which I like to hear from you. So thank you very much, sir.
See, I tell you, all the faculty members who are present here, we want to make our students industry ready.
Yes, sir.
And for making them industry ready, they should be exposed to all these requirements which are daily done in the industry. Unless we do that, they are not going to be accepted by the industry, and the industry will always complain to us that your students are not knowing the required knowledge.
Hmm.
So let us try to do something in that direction.
Actually, I want to I want to discuss that the uh, the reason behind to keep this webinar, sir. We have arranged one placement fair at our institute, and 4 institute students also came over here, and around 6 to 7 industries came. So one of the parental industry persons, they informed me that students are not having sufficient knowledge, actual knowledge of industry. So at that time only, I have decided
Okay.
to to keep one webinar regarding the updations in industry. So this is
I hope
yes,
this has helped them.
yes, sir,
Sure,
sure.
sir. Yes, sure, sir. It is the need of the time, sir, in current condition also.
Very true.
Okay. Because industry required immediately prepared material. Okay. Thank you, sir.
So thank you very much, sir, again for accepting our invitation.
It's a pleasure anytime. As far as I'm available, I can share my knowledge. Ultimately, whatever I have learned and whatever I am learning, it's nice to pass it on.
Yes,
Right?
sir. Thank you, sir.
Okay. Thank you very much, everybody.
Thank you so much, sir.
Right.
We can conclude now, madam. Thank you. Thank you, everyone.
Yes. Thank you. Thank you, everyone, students, faculty, other institute principal. And
We can close it, madam.
yes, sir.
Okay.
Thank you, sir.